Cool. I think we're live, guys. All right, hi. So I'm going to not stand in the screen this time. I can't, how do I tell if there's people seeing it?
It should say 4 hours.
We could get on our phones and share that.
Yeah, maybe. So just search my
name.
You guys, I'm a social media influencer. Great.
I know, I mean, I always like.
So if I put this in front of that.
Okay, come look at this, you'd like this.
Yeah, that's what it is.
Starting photo.
All right. So it's 11. If you're tuned in, hello. This is weird. I'm gonna draw on the whiteboard a lot today, so I will try to stay out of the way of that. I don't know. I feel like that's weird. I don't know. If you are following along live, actually, let's do this. I'll have it, sorry. Let's just do, it's kind of facing, so if you're following along on the live stream, I posted the PowerPoint slides to Blackboard. Sorry, I feel like I'm not talking to you guys. So you can follow along, and then when I use the board, I'll just turn the screen. So let's go ahead and get started. So just as a reminder, your guys' homework is here. So we are starting chapter 14 today, which is all about Mendelian genetics. And we're going to see that the information that we learned about meiosis, we're going to use again today, just in a slightly different version. Remember that meiosis makes gametes. And today we're going to take those gametes and see that we can make predictions about what the offspring are going to look like. So just make sure you get that done by Monday at 10:30. All right, so today with Mendelian genetics, this is so really for the rest of the semester, we're going to be talking pretty intensely about genetics. And I'm really excited about that because this is really what made me fall in love with biology. And so hopefully you guys come away with some appreciation for this topic too. And we're going to start kind of in the order that this was studied back in the ancient times. So we're going to start with Gregor Mendel and talk about how, without knowing what DNA was, without knowing what cells were, he was able to figure out how parents transmit their traits to the next generation. And genetics has been around for a really, really long time. Humans have long understood that traits can be passed down from parents to offspring. In fact, humans really have been doing this for millions of years. They've been manipulating organisms. For example, wolves, all domesticated dogs can be traced back genetically to wolves. So it started with cavemen or some ancient group of humans deciding, hey, this wolf seems more tame. It wants to hang out by our fire. Why don't we keep breeding it? And now we have everything from Great Danes to Chihuahuas. Domestication of plants has happened pretty frequently as well, where people take the food that they want to eat and they breed it and make it look different. Like corn that we eat now looks very different from corn that existed hundreds of thousands of years ago. And there were a lot of ideas that existed before Gregor Mendel came along to try to explain what was happening in organisms. So there are a variety of theories that existed in order to explain how traits were passed from one generation to the next. So it was pretty clearly known that we can look in this room and you can compare yourself to your parents and you can see I look a little bit like my mom or a little bit like my dad. So people have known for a long time that traits get passed. One of the first ideas to explain how genetic material gets passed from one generation to the next was this blending idea. Blending said that genetic material that's provided by the parents mixes, and that's why offspring look like a mix of their parents. However, we know that is not true because if blending was a real thing, all of us in this room would look pretty close to the same. We'd all have kind of this middle of the line skin color, middle of the line height. We might even have a weird gender where we're not fully male, not fully female. So we know just from looking at a population of people or organisms that blending doesn't actually explain why we have variation within our populations. Blending also doesn't explain why some traits can skip generations. On Monday next week, we'll talk about pedigrees, which allows us to trace traits in humans. And we'll see that some traits, like diseases like hemophilia, for instance, tend to skip generations, and blending doesn't explain why that happens. Another theory that existed was the particulate theory. And you can kind of think your book describes this as traits kind of existing like a deck of cards in a person. And when a person goes to make their offspring, they give just some of those cards to their offspring. And so because of that, you have the shuffling of cards. So your offspring are receiving these particles that make them slightly different from you, for example, as a parent. And so this was saying that parents pass on discrete heritable units. Remember, heritable just means that you can inherit that thing. And those units retain their full characteristics in the offspring. And we'll see that that's not always the case either. And so the particulate theory, it's a lot closer to what actually happens, but it's not the exact right situation. And so we had this blending theory, we had this particulate theory, and then Gregor Mendel came along. He has a really cool history. He started out as a monk, and he was just kind of breeding pea plants at his monastery and learning, oh, look, if I breed these two together, I get a slightly different type. And he really just started by having an interest in in breeding plants. And he really was instrumental in creating what we now know as modern genetics. And so we're going to talk a lot today about his experiment. One of the big key things about Mendel is that he really took the time to perform controlled experiments. He did hundreds of replications of all of his experiments. He had huge amounts of data that he could then analyze. And he very much ensured that everything he did was very controlled. So he truly was a true scientist in the sense of how he did these breeding experiments. We're going to use this figure a lot today in class, and we'll see that there's a lot of information just in this one figure that we'll break down. When we talk about Mendel's experiments, they're just like our meiosis lecture. This is a pretty vocabulary heavy lecture. And I've listed all of those in your lecture outline. So you don't, if you're writing your notes from scratch today, you don't have to write all of these down on the board. We're also going to talk about them a lot as we go through, so you'll be able to define them in the other parts of your notes as well. But just know if you're making flashcards, you can refer to this list here for the key vocab words you need for this lecture. Again, at this time, we did not know about DNA. So we didn't know what was the actual thing that was being passed from one generation to the next. But they did know at the time that there were characters. So for instance, a character is just a heritable feature that can vary among individuals. So a character can be, if we're talking about pea plants, which we'll really focus on today, that could be the general flower color of that plant, or the peas themselves can have a texture. They can be either wrinkled or smooth. The character is like the general category that we'll be talking about. Within each character, there are different traits. So for instance, flower color can be purple or white. Pea pod shape can be smooth or wrinkled. So the traits fit under the umbrella of the character, and it's the traits that are the things that make each offspring different. We're going to talk a lot about true breeding. So Mendel, he always set up his experiments with what are called true breeding, or what we would now call purebred plants. He knew that, for instance, he had purple plants, and generation after generation after generation, those plants always made purple flowers. So he knew that they had only that trait. Their character of flower color was always going to be purple. He also had true breeding white flowers that we'll talk about here in a second. And they always produced white flowered plants. So true breeding just means generation after generation, the same traits will stay as long as you breed two true breeding parents together. And then he took these purebred or these true breeding plants and he would mate them together, and he called that hybridizing. So he knew that if you took a true breeding purple and a true breeding white plant, they would be hybrids. And so hybridization just says that you're taking two different purebred plants, purple and white, for example, and you're making a hybrid. For P generation, F1 generation, F2 generation, and alleles, I'm going to move on to this picture so that we can see in a visual way how these terms come in. And I'm also going to use the board. So in Mendel's experiments, he always started with what was called the P generation. I'm just going to make sure that you guys can see this in the video real quick. Cool. Oh, is it backwards though? I don't know. There's only 3 people watching, so if it's backwards, I'm sorry. That says P generation. All right. And you can think that P stands for parent. Our parent generation is always going to be true breeding. So he's always going to take true breeding plants. So these are true breeding always. And if you're looking at the picture on the PowerPoint, he specifically took two pure breeding plants that were the opposite colors. So he mated a purple plant that he knew came from a true breeding line, and he crossed that or mated it to a white flower plant that he also knew came from a true breeding line. So he knows that if you were to take a purple and a purple, you would always get purple offspring. If you were to take white and white, you would always get white offspring. After he crossed these parents, this made our next generation. That first generation is called the F1 generation. F stands for filial, F-I-L-I-A-L. You can also think of it as family. So once you have offspring, often people refer to themselves as a family, right? And so this is the first generation of that family. And he knew that all of these offspring were going to be hybrids because he took true breeding plants of the opposite color. So we'll see that we are always going to refer to the offspring that come from true breeding parents as hybrids. But he got a pretty interesting result that he didn't expect. Every single one of the plants that came out of that F1 generation were also purple. So that was weird. He said, I have a purple parent and a white parent, but now I have only purple offspring. We're going to see why that happened here in a second. But he's like, okay, interesting. Not what I expected. From here, he then decided, well, I'm going to take 2 of these hybrids from this F1 generation, and I'm going to cross them together. So he took 2 of these purple plants from the F1 generation. It's a good thing this is plants and not people, because you'd be causing siblings to mate if this is people. But he took these 2 purple hybrids, he allowed them to mate, and when they created offspring, they created what's called the F2 generation. So again, second filial generation or second family generation. These guys are also hybrids because they came from hybrid parents. And he saw something really weird here. Again, not expected. He saw that out of this F1 cross, when he crossed 2 purple hybrids, 3/4 of them were purple, but 1/4 of them were white. So that second trait came back up again. So this was really surprising to him. He was like, wait a second. So first off, one of these traits is completely hidden in the first generation, but it comes up again in the second generation. What is happening here? But it's really these observations that then led him to further study this. He repeated this experiment hundreds of times. He did this not just with flower color, but with pea pod shape. Peas have different heights, so he would take like tall and short and mate them together. He would do this for pea pod color. The peas can be green or yellow. So he took all these different traits and did this same experiment where he started with pure breeding plants, got an F1 generation. Those F1 hybrids were crossed to one another. He got an F2 generation, and he always saw these ratios. He always saw that in the F2 generation, you always have 3/4 of those offspring showing one trait and 1/4 of those offspring showing the other trait. And so because of that, he came up with his first law of genetics, which is called the law of segregation. And so for those of you watching, I'm going to flip it back to the PowerPoint. Oh, also, sorry, was I standing in the way of that? There. So that's what we drew on the board. I'm not good at this, guys. Oh, cool. It's not backwards. Sweet. Thanks, Kevin and Sarah. Oh, sorry. All right, so that's what we drew on the board. And it's just matching the picture that we just had up on the PowerPoint. So we're going to go through what Mendel's laws were, and his first was the law of segregation. In his law of segregation, he's only looking at one trait at a time. So notice that in this, we're just looking at flower color. We're not looking at multiple traits per experiment. But from crosses like this, from these experiments, he always saw that the F1 generation always only showed one of the two parental traits. So purple or green or wrinkled. And then the F2 generation always came up in this ratio, 3/4 purple to 1/4 white. And from that, he created this law of segregation, which has some rules that we're going to break down on the next PowerPoints. So of the law of segregation, his first rule to explain why he saw these patterns arise, he first thought, okay, there must be alternative versions of genes or traits that account for variations in the inherited characters. So he said, to be purple means that this parent has different traits than this white parent. So he was saying to explain why you see differences, that each parent or each offspring, every single individual must have these different traits. We now know that those traits are genes. At the time, he didn't, but because we know that now, that's why I put in genes here in the slide. The different versions of genes have a name, and they are called alleles, A-L-L-E-L-E-S. So the purple trait for our flower color, that is one allele. That is one version of the flower color gene. And white is the second allele. That's the other version of the flower color gene. So alleles just tell you that you have, for every gene, you have multiple alleles. Sometimes there's only two versions, only two alleles. Sometimes there's more than two. It just depends on that gene. He got really lucky in that most of the traits that he decided to look at only have two alleles, like purple versus yellow, smooth versus wrinkled, things like that. We now know that alleles exist because the information in that gene is slightly different. So overall, remember, we talked about how on Monday, every single person has 2 copies of all of their chromosomes, and those chromosomes contain genes. On those 2 copies, they can have 2 of the same alleles. In that case, they would be true breeding, or they can have different alleles, purple and white. In that case, they would be considered a hybrid. And so we know now that every gene can have a slightly different set of instructions. Overall, it's still controlling flower color, for example, but you can make purple versus white color just because that gene carries a slightly different set of instructions than between the 2. So that was his first rule, that there must be different alleles that explain why you're seeing different traits in these characters. In the law of segregation 2, he also said that for every character, so flower color or pea pod shape, or height or gender, if we're talking about humans, that organism gets 2 alleles. It inherits 1 allele from one parent and 1 allele from the other parent. So again, kind of tying back to meiosis, remember, we're going to split chromosomes apart. So every gamete just has 1 allele of that gene. But then when a sperm and an egg come back together, we get now 1 allele from one parent and 1 allele from the other parent. And again, those alleles can be identical, so that if the 2 parents have, for example, if we look at this little family tree, we have a mom, and she has 2 blue alleles, because every organism has 2 alleles of every gene. But dad has different alleles. He has 1 brown allele and 1 blue-eyed allele. In this situation, all of their children got identical alleles from both parents. They each received a blue allele from both parents. It could have happened if they have a 4th child, for instance. This dad could give his brown allele, and then in that case, the child would have a brown and a blue allele. But so the alleles that offspring inherit can either be identical, as is the case in this family tree, or they can be different. And we'll see that difference here in a moment. And the thing to remember here is that in our somatic cells, the cells that make up all of the parts of our body except for our reproductive tissue, all of them have 46 chromosomes total, or 23 pairs, because one of those sets came from one parent, the other set came from the other parent, as we talked about on Monday. And so because of that, for every gene that we have 2 copies, and potentially 2 different alleles for every single gene. Okay, so if we go back to our picture, we know that our true breeding parents, so true breeding, again, we know now that they have 2 alleles, and because they are true breeding, we can say that those alleles for the purple parent must both be purple. So true breeding means that their alleles are identical. If our white parent was true breeding, we know that it must have 2 white alleles, because again, to be true breeding means you have identical alleles. And now what we're going to do, we know that these offspring in both the F1 and the F2 also must have 2 alleles, but Mendel needed to think about, why are we only seeing purple in the F1 generation, and why does white show back up in the F2 generation, but at a pretty low frequency compared to that purple color? So for him to explain that, he said that if you have 2 alleles at a locus, remember locus is just where a gene is located on an overall chromosome. If you had 2 alleles at one locus, and if they differ, so remember in the hybrid plants, these guys are a mix of purple and white, so they must have 2 different alleles. If those alleles differ, one of those alleles can be considered dominant, meaning it will always show through, while the other allele must be recessive, it can be hidden. And so he gave a name to these different alleles. You can have dominant alleles or recessive alleles. And so to be dominant, it's always essentially going to win. And to be recessive, we'll see that it can be hidden sometimes, that trait can be hidden. So if we just go back to our original drawing, what trait, purple or white, is hidden? White, yeah. So in the F1 generation, we don't see white. And so we now can also say that the white allele must be recessive because it's hidden. What about the purple allele? Because we always see it in every single generation. What can we say about the purple allele? It's dominant. It's dominant, great. What's nice about this is it now allows us to annotate the actual versions of the alleles that these organisms have. Usually, the rule of thumb is that capital letters, so to talk about alleles, we always use just a single letter. Capital letters denote that that allele is dominant. Lowercase levels tell you that that allele is recessive. So we can actually say what versions of what alleles these parents have. So for example, if we know that we have a purple parent that is true breeding, we know that it has 2 copies of the dominant allele. And let's just use the letter P. And I'm using a capital P there to show that this parent has two versions of that gene. They are identical. They're both big P, big P. This white parent has the recessive version, so we're going to use lowercase letters. We're going to use the same letters as what we used over here. We also know that the white parent has the recessive trait, and so we can say that because it's pure breeding, we know it has two copies of little p. If letters are hard to distinguish between whether they're capital or lowercase, I just put a little line on top to tell you that that's a little p. The other thing you can do is draw like a cursive version for lowercase letters, but sometimes cursive versions look similar as well. So I put lines on top of my little p's to tell you that that white parent has two little p alleles. The rule about recessive traits is that the only way a recessive trait can show itself is if that individual has two copies of that recessive allele. So notice that this parent is white because it only has little p alleles, so it has to show that white trait. Okay, so at this point, we know that every organism has two versions of every single character. So in this case, we have two alleles for every trait that we are trying to discuss. We also know now that one of those traits is dominant. It will always show itself in every generation. One of those traits is recessive. It's hidden in the F1 generation, and it will show itself again in the F2 generation. So you always can look at the F1 generation to figure out which of your versions is recessive. We now know too, because of dominant versus recessive traits, we can actually annotate the actual versions of the genes that these guys have. Big P for our true breeding parent, little p for our true breeding white parent. The last step that Mendel had to do was to explain what's happening in the F1 generation. And to do that, he had to think about meiosis without knowing what actually is happening in meiosis. So again, kind of crazy, but Mendel did not know what cells were yet. They didn't know that gametes made new organisms, but he said that something must be happening to those two alleles in the parent. He said specifically those alleles must be segregated. This is where the law of segregation gets its name. Those alleles must be segregated in order for offspring to get one allele from one parent and one allele from the other parent. So again, remember that we talked on Monday. So for instance, in this cell here at the top of this picture, for those of you watching on YouTube, I'm circling this top circle here. Remember that every cell starts out as diploid. In this parent, this cell, for the T trait, that parent has one big T and one little t. So that homologous pair is big T, little t. It has two he or she, this parent, has two copies of this gene. Remember that in meiosis, in prophase 1 of meiosis 1, those homologous pairs find each other, so the big T and the little t line up. But then remember at the end of meiosis, once we get through meiosis 2, those homologous pairs are separated so that the big T goes into one gamete and the little t goes into the other gamete. So Mendel said that during gamete formation, these two copies of our alleles must get separated. So the law of segregation says that we all have two copies of every single trait. Of those traits, one is dominant, one is recessive. And during gamete formation, those two traits must get separated into their own gamete, which is being again shown here. Big T is going to one gamete, little t is going to the other gamete. And so we can now explain the hybrid situation for our F1 generation. So again, if we go back to our parents, when these parents make gametes, they're going to end with two gametes in this situation. And we're going to split this pair apart. So big P on the left is going to go into the gamete on the left. Big P on the right will go into the gamete on the right. So this parent will always make gametes that have big P. The white parent, its alleles will also get separated. So the little p on the left will go into the gamete on the left, and the little p on the right goes into the gamete on the right. So these gametes again are going to meet up during fertilization. So a big P gamete from one parent and a little p gamete from the other parent will fuse. So what letters must our F1 generation have? Big P, little p, great. Big P, so their cells all have one big P and one little p. So they have to get big P from one parent, little p from the other parent. We know that big P is dominant. That trait is always going to win over our recessive allele. And that explains why all of these organisms in the F1 generation are purple. They all have one copy of that dominant gene. You only need one dominant in order for it to show itself. Okay. When we get to the F2 generation, we can do what's called a Punnett square. Before I show you how we can figure out these 3/4 and 1/4 ratio though, there are some terms to understand before we get there. So when we want to talk about the trait that we see versus the two alleles that an organism has, there's some different terms to keep in mind. So first off, we can talk about the condition of the alleles that an individual has, and those can be called either homozygous or heterozygous. Remember, homo means same, hetero means different. Homozygous is referring to the fact that both genes are identical. So for instance, our big P parents, our little p parents, they are considered homozygous because they have two copies of the same version, that same allele. Another way that you can refer to the homozygous condition is we could call those parents homozygotes. So you can say homozygous or homozygote. Hetero, again, means different. So a heterozygous organism or a heterozygote is an organism that has different versions of each allele. So for instance, our F1 generation would be considered heterozygous because they have one big P, one little p. They have the two different versions. So we can add the terms homozygous and heterozygous to our picture now. If we want to talk about the genes that an organism has, so saying big P, little p, big P, little p, we are talking about the actual versions of the genes that organism has, and that's called the genotype. So whenever you want to talk about the genes, that's the actual alleles that an organism has, we are talking about the genotype. So we could say that the genotype for our purple parent is homozygous dominant. It has two copies of the dominant allele. Or we could say the genotype of the purple parent is big P, big P. We could also say that the genotype of the white parent is what? Homozygous, and specifically homozygous recessive, because it has two copies of the recessive gene. And then we can just say that our F1 generation, their genotype is heterozygous, and we automatically know that that means that they must have one dominant and one recessive allele. So genotype tells you the actual letters that exist for those genes. Phenotype shows you tells you about the trait that you actually see. So phenotype for us would be the actual flower color that we see in these parents and offspring. So we have two phenotypes for these flowers. What are they? Purple and white, yeah. So genotype are the genes that you have. Phenotype is what is the actual trait that you see once those genes are expressed. Okay. So now that we know these terms, we can see what Mendel did in order to figure out why you always saw that in the F2 generation, 3/4 of them would have the dominant phenotype, whereas only 1/4 of those F2 offspring have a recessive phenotype. So what we see, whenever we talk about Punnett squares, we're always talking about, we're using the genotypes to figure out the ratios of the phenotypes, and we'll break this all down. So I'm gonna erase this so that I can use the board. Are you guys good with that? Mm-hmm. Okay. I'm gonna go to the YouTube. Are you okay? The YouTube, like an old person. Can I erase this? Can someone type yes? Okay, I'm gonna erase it. No one said anything, so that's a yes. All right, so for you guys, I have given you blank Punnett squares in your lecture outline. We are first going to do a Punnett square for what is called a monohybrid cross. So you guys have just empty grids that look like this in your notes. So this grid is called a Punnett square. It's named after the guy who invented this. So Mendel actually didn't invent the Punnett square. Punnett did. And we're gonna first look at a sorry, I'm gonna use a different marker so you can see it better. Oh, that one's not any better. We're gonna do a monohybrid cross. What does mono mean? One. Yeah, so we're just meaning, when you hear the term monohybrid, we're just talking about one trait. So for instance, we're just gonna look at purple right now, the purple flower color trait or white flower color trait. So monohybrid means one trait. And we're specifically talking about hybrid organisms. And so the parents are hybrids, meaning they are not pure breeding. They have a combination of alleles instead of having two of the same. So remember that to get to the F2 generation, Mendel took his F1 plants. So remember that in that F1 generation, plants are big P, little p. That's their genotype. And he crossed two individuals from that F1 generation together. So we can say that he took two heterozygous individuals. They both had the genotype of big P, little p, and he crossed them together. In order to figure out our Punnett square, what goes on the outside is our gametes. And so we need to figure out what gamete combinations can be formed from both of these parents. So remember, right now, as we've written this is the homologous pair of chromosomes. And that homologous pair gets split up in meiosis II. So we can say that when we create gametes, some of those gametes will be big P, the other set of gametes will be little p. So you always have to start with haploid on the outside so that we can get diploid on the inside. Yeah.
So outside are like what the gametes that we get when we separate them.
Yep, yeah. So yeah, we have to figure out from these parents now what gametes can they create. What gametes, so if this is parent 1, what gametes is parent 2 going to make? The same, yeah. A big P gamete and a little p gamete. Okay. These individual big P's and little p's get written on the outside of our Punnett square. So I'm going to take parent number 1. I'm going to put their big P here and their little p on the bottom row. And then I'm going to take the other parent number 2 and put their big P on that top, on the left-hand column, and their little p on the right-hand column. So now we know that these parents are going to make these gametes. Everything we fill in inside the Punnett square is the F2 generation. So we can now figure out the genotypes of the F2. And the way you do that is you literally just read rows and columns. So for instance, if I take my big P's and I drag them in, this offspring is going to get one big P from one parent and another big P from the other parent. So its genotype is big P, big P. What's this top right box going to get?
Big P, little P.
Big P, little p, great. Is this homozygous or heterozygous on the left? Homozygous, great. And specifically, is it homozygous dominant or homozygous recessive? Dominant, great. So we know it has two capital letters, so it's homozygous and it's homozygous dominant. What about this guy? It has one version of each.
Homo and
Not homo,
hetero.
hetero now. So heterozygous. And again, we don't have to say dominant or recessive because they have one dominant and one recessive. So this is homozygous dominant. This is heterozygous. And now see if you can fill out the rest of this Punnett square. What should the bottom left genotype be?
Big P, little P.
Yeah, big P, little p. So we always put the capital letter first, just FYI. So this guy is also heterozygous. And then what's the bottom right box going to be? Little p, little p. And how could we describe that genotype? Yep, homozygous, recessive. Perfect. Cool. So filling in the Punnett square tells us the genotypes, but we don't know yet the phenotypes. So remember that there's only two phenotypes we can see. What are they? Purple and white. And remember that to see the purple trait, an organism only needs to have at least one big P. So we can say that the purple phenotype is caused by a genotype that has at least one big P, because it technically doesn't matter what is in that second position. We can just draw a line, and that represents that this second position could be another big P, or it could be a little p, but in either situation, that will give you a purple flowered plant. What is the only way to be white? What genotype do you have to have? Two little p's. So these guys have to be homozygous recessive. So we know that the only way to have white flowers is to be little p, little p. So how many total offspring variations can we get? How many boxes do we have? 4. So we can say that out of 4, we're going to see certain ratios. So we're going to make the bottom half of our fraction, the denominator, is out of 4 because I have 4 total boxes inside my Punnett square. Out of that 4, how many will be purple? 3. Yeah, so big P will be purple, and big P, little p will be purple. And so 3 out of my 4 are purple. How many will be white? Just the 1. So just my little p, little p. So notice that by figuring out a Punnett square, we can first figure out the genotypes of all the possible genotypes that can be made from this cross. And we can figure out now the phenotypic ratios. In a monohybrid cross, when you're looking at one set of hybrid parents, so look, we're just looking at one trait here, just flower color. In a monohybrid cross, if both parents are heterozygous, you will always see this situation where 3/4 will be the dominant phenotype, 1/4 will be the recessive phenotype. Do you guys have any questions before I give you a practice question?
Well, I mean, just if parents have multiple generations, they all technically, in theory, could be 2 small peas and then the next generation. I mean, we're talking about peas, so they have a lot of seeds, but if we're talking about a human, the chances are they could have.
Yeah. So for instance, if this was people, because people usually have one kid at a time, this allows you to figure out that if 2 parents were heterozygous and they mated, you could say that they have a 3 in 4 chance of their child showing the dominant phenotype, or they have a 1 in 1/4 chance of their child showing the recessive phenotype. So now that what's cool is that now that you have these, you can work with them to do to make predictions about total numbers. Taylor, I'll come to you in a second. So for instance, if we had 100 offspring total, how many of those would be purple? Yeah, how did you guys get that? Yeah, we know that of that 100, 3/4 of them should be purple. And so 100 times 3/4 is 75. So we should see 75 purple plants. How many should be white? 25, yeah. So again, we know out of those 100, 1/4 of them should be white, which means that 25 of them should be white. And again, if you can check your math by adding those together and it gets you back to 100.
Can you say 75 would be dominant, 25 are recessive?
Yeah, you can say that too. 75 would show the dominant phenotype, 25 would show the recessive. Taylor, what was your question? Yeah. So if you're talking about, I'm going to teach you guys another trick on Monday. So we're going to practice some monohybrid Punnett squares here, and then we're going to do a harder type of Punnett square, and they take longer. So I'm going to teach you guys some math tricks on Monday to do that faster. But yeah, for these, we're looking at genes that just have 2 versions. Yeah, that's a good question. Okay, so I'm going to give you guys another practice problem. So I'm going to give you, let's do the letter B, because it's a little bit easier to see when it's capital and lowercase. So I'm going to tell you that there is a big B allele. I'm going to use red again. And the big B allele gives brown fur color. And the little B allele gives white fur color. I want you to cross 2 heterozygous parents. If they are heterozygous, what must be the genotype?
Big B, little b.
Big B, little b. So I want you to take big B, little b, crossed to big B, little b. And I want you to figure out the Punnett square and then give me the phenotypic ratios.
You said those are hetero?
Yep, so these are 2 heterozygous parents because they have 1 version of each allele.
Wait, what do you want us to do?
So I want you to figure out, using this, I want you to figure out the Punnett square. So what are the genotypes, and then list out the phenotypic ratio. So I'll give you guys a couple of minutes on that. And you should have a second blank box in your lecture outline that you can use. On the next page, so you can also use known genotypes of children to figure out what genotypes the parents have. So if you flip the page, you'll see that the inside of your Punnett square is filled out, but the outside is not. You can try that one too. So on the outside of your big B, little b Punnett square, you should have that your top row is big B, the bottom is little b, and then big B on the top left, little b on the top right. So you should have gotten that the top left is big B, top right is big B, little b, bottom left is big B, little b, and bottom right is little b, little b. Did you get that? Yes. Cool. What are the 2 phenotypes that we can see from this? What 2 fur colors? Yeah, so we can get brown fur or white fur. So we know that some of our offspring are going to have the phenotype of having brown fur. Some will have the phenotype of having white fur. How many will be brown furred? Yeah, so anything that has at least 1 big B. And how many have white fur? 1/4. What genotype do you have to be white? Little b, great. Perfect. Cool. And then, yeah. So the next Punnett square, and I forgot my lecture notes. So I'm going to keep going. Yeah. So if you turn the page, I filled in the inside of the Punnett square, and you can use the inside to figure out the parents. So in that Punnett square, you have big Y, big Y, big Y, little y, big Y, little y, little y, little y. So now see if you can figure out what must the genotypes of the parents be. And you can also try that for the other Punnett square that has R's in it on the next page. Or sorry, the next box. So again, if we work backwards, if we take these big Y's and put them out here, we know that what letter must go on the top. Yep, so we know that this must be big Y and a big Y. And then to have anything that has little y's in it, and especially to have little y, what must both parents also have? What allele? Little y, little y. So you can work backwards too. So now we know that the parents were big Y, little y, big Y, little y. So see if you can figure out those R's. So for the next Punnett square, am I going too fast? Okay. For the next Punnett square, all of those offspring are heterozygous for big R, little r. So see if you can figure out what must the genotypes have been of the parents. Yeah.
For the exam, does it matter if we put like so for this one it'd be 2 big R's and 2 small R's? Does it matter if we put the 2 big R's on the top or the side?
Nope, as long as you just get their genotypes right. So you're right that for every single, so notice that this looks like the parent to the F1 generation. So this tells you that one of the parents has to be homozygous dominant. The other parent has to be homozygous recessive. You can put the little r's on top or on the side, doesn't matter the order, you'll still get the same answer inside. So we know that these parents must be big R crossed to little r.
Let me leave that up to one side.
Yeah, for sure. Yeah, sorry, I know I moved kind of fast on that. Do you guys have any questions? I put an entire practice worksheet under study aids and the key as well. I highly recommend just doing that worksheet over and over again. It'll help you out a lot. The key is also posted.
This might be stupid, but for the test, should we know better off to label like the full thing by ourselves and knowing homozygous, heterozygous, recessive, and dominant?
Yeah, so for the test, you will definitely need to know how to do monohybrid Punnett squares.
Okay.
Because that's going to help you get answers. We're going to do some practice problems on Monday next week. That you'll see that we're going to use the information that comes out of the Punnett squares to answer kind of like a second level of question. So know how to fill out Punnett squares. Know terms like homozygous dominant, homozygous recessive, and heterozygous.
Okay.
And know like phenotype versus genotype. So phenotype's the trait that you see, like brown fur. Genotype's the gene combination that gives you that phenotype. So oftentimes, and this happened with Gregor Mendel too, was he would do crosses. So he would have, so notice that when you have a dominant phenotype, you can't tell if they are homozygous dominant or heterozygous. So if you have a organism that you know it's showing the dominant trait, but you don't know what its genotype is, you can do what's called a test cross. And a test cross allows you to figure out whether that dominant phenotype has a homozygous dominant genotype or a heterozygous genotype. So when we perform a test cross, we're using the offspring to figure out if the dominant trait from the parent was caused by the homozygous condition or the heterozygous condition. To do this in a test cross, so we're going to have a parent where the second allele version is unknown. So notice that in this purple flowered parent, we have just a big P and then a dash because we don't know if it has another second big P or a little p. But to do a test cross, the second parent always has to have the recessive trait because you know to have that recessive trait, that genotype has to be homozygous recessive. And there's two combinations that you can get out of this which help you figure out the parent. And it basically is doing the same thing that we just did with working from the inside of our Punnett square to the outside. So notice that in this figure, if you have a situation where all of your offspring are purple, that tells you that you know that those offspring, so for instance, we know that one of our parents has to be little p on the outside of our Punnett squares. We know that the other parent has at least one big P, but we don't know what's in that second position. If you cross this parent and you don't know its second situation to a homozygous recessive parent, if you get a situation where all of the offspring are showing that dominant trait, so all 4 out of 4 offspring are purple, we know that they have to have at least one big P in order to be have that dominant phenotype. And we know that the white parent can only give that little p allele. That's all it has to give. So we know that every single one of those purple offspring must be heterozygous. So that tells you that the unknown allele in the purple parent is what letter? Big P, yeah. So it has to be big P in order for every single organism, every single offspring to have a big P and a little p. If you saw the reverse, so if you saw the situation on the right-hand side of that cartoon up top, if you saw that half of your offspring were white, you know that they have to be little p to be white, right? And then you saw that the other half were purple, and we know that they had to get that little p from one parent, and to be purple, they had to get the big P from the other parent. If you have where 2 out of 4 are dominant and 2 out of 4 are recessive, that tells you that the unknown allele has to be what letter? Little p, yeah. The only way that you can have little p offspring is if both parents can give a little p allele. So sometimes you don't have to know the genotype. You can use the offspring to work backwards. So the whole idea with that is that. So in the last 15 minutes, so at this point, we've only looked at one trait at a time, just flower color. But Mendel also, he wanted to look at multiple traits at a time. So he wanted to say, so if you think about a pea plant, they can be tall and have purple flowers and have wrinkled peas. They have multiple traits, right? All of us have multiple traits as we look across this room. And so he made all the law of segregation based on just looking at one trait at a time. So he performed monohybrid crosses. He just looked at flower color or pea pod shape, or we looked at fur color for our big B, little b example. But he knew that, and we know that organisms have just more than one trait. And so he wanted to follow 2 characters at a time. So he wanted to look at, say, flower color and pea pod shape. And so to do this, he had to perform what's called a dihybrid cross. So di means 2. So dihybrid, what we're going to look at now, means that we are looking at 2 traits, and the parents are hybrid or heterozygous for both of those traits. So for dihybrid crosses, we now have 2 traits, and the parents are still hybrid for both of those traits. When he did dihybrid crosses, when Mendel looked at 2 traits at once, he developed his second law, which was called the law of independent assortment. And what he said here was that the 2 alleles of the 2 genes, they sort into gametes independently of one another. We're going to see how that works here in a second. And we're going to see that in these Punnett squares, we are always going to get a phenotypic pattern of 9 to 3 to 3 to 1. So we're going to see how this works here by actually doing a Punnett square. So you guys in your notes have a Punnett square that is a 4x4 grid. So I'm just going to draw that on the board here. Cool. I'm just going to check with the, cool. All right. So we're going to do a dihybrid cross. So in this situation, we are going to get a Punnett square that is 4 by 4. How many total squares are inside that Punnett square? 16, yeah. So there are 16 possible offspring genotypes that we can get from a dihybrid cross. So we're going to look at, we're going to look at pea color. So the dominant color for peas is yellow, and we're going to use big Y to represent that. The recessive pea color is green, so we'll use a little y to represent green peas. The dominant shape or texture is round, so we're going to use a big R to represent peas that have that round shape, and a little r is going to represent wrinkled. It's recessive. So first off, remember that gametes have to go, so ultimately what we're going to do is we're going to take two parents that are heterozygous for both of these traits. That means that their genotypes are big Y, big R, little r. The only thing that is tricky about a dihybrid cross is figuring out the gametes, and you can use foiling to do that. So remember that these Ys have to separate when we make gametes, and Rs also have to separate. And we're going to see that Ys and Rs separate from each other without, they don't care what the other one's doing. So Ys will split in half in any potential order, and it doesn't matter which R also splits with them. So to show you guys that, for instance, so we're going to get 4 different gametes from this situation. So I'm going to draw bigger circles, but I'm going to draw 4 circles to represent the gametes that we can get out of this situation. So first off, we know that every single gamete will have one Y and one R. For instance, this big Y could go with this big R, and so we could get gametes that are big Y, big R. Or because genes assort themselves or sort themselves independent of the other, that big Y could go with that little r. So we would get a gamete that has big Y, little r. The little y, I'll use a different color for the little y. Little y could sort with big R, so we would get a gamete that is little y, big R. Or little y could sort with our little r, so we could get a gamete that is little y, little r. This is the trickiest part, is making the gametes. I think foiling is the easiest way. So the first rule of your gametes is remember that they only have one of each letter, so one Y, one R. The big and little letters can sort independently of each other. So you could get a gamete that has both big or a mixture of big and little, or gets both littles. And then those gametes, again, are going to go on the outside of our Punnett square. We have 2 parents that are this genotype, big Y, big R, little r. So both parents are going to make these same 4 gametes. So for instance, that big Y, big R can go on that top row. And then I'm just going to fill in big Y, little r is the next one, little y, big R, and then little y, little r. So those gametes go on the outside. Same set of gametes goes on the top. Big Y, big R, big Y, little r, little y, big R, little y, little r. So what we're going to do when we fill in our Punnett square is Y's are going to come first. Capital is always before the lowercase, and then R's are going to come second. So in this top box, What Y combination does this offspring get?
Big line.
Big Y, great. And then what R combination?
Big R, big R.
Big R, big R. Cool. What about the next square over? What Y's does this offspring have?
Y, big Y.
Yep, 2 big Y's, big Y, and then its R combination is
Big R.
big R, little r. So just see now if you can finish filling in that Punnett square. So for instance, the next one is big Y, big R, and then big Y, big R, little r. And then just keep going down that row. Notice that this process does take some time. You can always use this process on a test, but I'm going to show you guys some quicker tricks on Monday. And I'll get out of the way for the camera here in a second. And then little y, big Y, little r. Okay. So your Punnett square should look like that when you're done. And then I'm just going to up the screen for right now so I can use the right-hand side. Just make sure the camera, okay, so the camera. Cool. All right, so we have figured out our Punnett square. We've filled it in. If you're still working on it, that's okay. Just keep filling it in. Again, how many total boxes are there inside our Punnett square? 16. So we're going to see that there are 4 different phenotypes that we get out of this. And all of our fractions are going to be out of 16 this time instead of 4. So for our phenotypes, we can have both phenotypes be dominant, which would be yellow and round. So we know that some of our peas will be yellow colored and their texture is round. Again, cool, you can see that, I think. We can also have some that are yellow but have the recessive texture. So they can be yellow but wrinkled. So we're done with our yellow color. We know that we can get green color, but the dominant texture, which is round, so green and round. We also know that we can get organisms that are both recessive phenotypes, green and wrinkled. And I'm actually gonna move this just so you guys can see it better. Just gonna plug it. Cool. So all of our fractions are gonna be out of 16. So we can put 16 here to start. But let's think now, to be yellow and round, what 2 alleles do we have to have? So to be yellow, you have to have at least one big Y. Great. So we know that anything that has at least one big Y will be yellow. So I'm putting a dash in the second place because the second position doesn't matter. What allele do you have to be round?
One big R.
One big R. And again, whatever's in the second place doesn't matter. And again, so for yellow wrinkled, we know that to be yellow, you just have to have the one big Y. What do you have to have, though, for your genotype to be wrinkled? 2 little r's, great. So little r, little r. What about green? What genotype do you have to be green?
2 little Y's.
Yep, little y, little y. And again, round can be at least just one big R. And then what do you have to be green and wrinkled?
Little y, little r, little r.
Perfect. Little y, little y, little r, little r. So now all we need to do is we need to count how many inside our Punnett square have at least one big Y and one big R. I'm gonna circle those in green. So if we go back to our Punnett square, in the top left, that guy has at least one big Y and little one big R. In the next box number 2 also has at least one big Y, one big R. This whole row fits that yellow round category. And then in the second row, boxes 1, 3 are big Y, big R. In row 3, we have box 1, 2, and in the bottom row, we have just box 1. So if you count up our total number of circles, how many did we just circle? 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 out of 16 are going to be yellow and round. To be yellow and wrinkled, you have to have one big Y and at least two little R's. Let's star those. We have none in row 1. Do we have any in row 2?
Yes.
Yep. Box 2 and 4. What about in row 3? Nope, nothing in row 3. And then anything in row 4?
Yes, just.
Yep, just the one right here. So we starred 3. So 3 out of our 16 will be yellow and wrinkled. For green and round, we're looking for 2 little Y's and at least one big R. I'm going to box those in blue. In row 3, we have 2. And in row 4, we have just the 1. So that ratio is also 3/16. How many are green and wrinkled? Just the one, just our one little guy down here in the corner. Special, yes. 1 is the loneliest number. So from there now, this is where the 9-3 to 3-1 ratio comes from. Notice that because the denominator, the bottom of our fraction is the same, we can just ignore it. And so we can say that 9 organisms will be the dominant phenotype for both traits that we're looking at. 3 will be 1 recessive trait, 1 dominant. 3 will be the other set of 1 recessive trait, 1 dominant trait. And only 1 will be the both recessive traits. We're going to learn some tricks on Monday. So again, I posted a practice worksheet for dihybrid crosses as well onto Blackboard. And the key for that is also posted. So please check those out. It's under the study aids section. So please make sure to check those out and practice them. Dihybrid crosses take a really long time. You can also look at 3 different traits, and that dihybrid cross becomes huge. Or sorry, that it'd be a trihybrid cross, and your Punnett square gets really, really big. So we're going to see that there are tricks that we can use on Monday. But I definitely recommend practicing monohybrid and dihybrid Punnett squares just so you are familiar with them. And that is all I have for you guys today. So have a great weekend. Stay warm. I'll see you on Monday.
On the dihybrid stuff, as far as you have the capital R is going second, so you can do that in dihybrid but not monohybrid. Because in the monohybrid, you had showed where the capital letter goes first.
Oh, so of that one trait that you're looking at, you always write capital first. Did I not do that somewhere? So like for Y's, there should always be the capital Y written first and then the lowercase, and for R's the same thing.
I think she means like you have little y and big R on the outside.
Oh.
Do we have this?
No. So notice that I'm keeping it in the order where Y's are coming first, R's are coming second. So I just kept it in that order. So no, you don't have to flip it around on the outside.
Thank you.
Cool. Thanks for checking in, guys.
Do we need to know how to do the dihybrid for exam?
Yes. Yeah.
Could R come first?